Good morning, everyone, or early afternoon, I guess. We are just going to give it one more minute to make sure everybody has a chance to get into the room today, and then we'll get started.
Good afternoon, Casey.
Hi, Zoe.
How are you? Yeah. How are you doing?
Doing okay. Doing okay. We're going to just give it one more minute and let everybody else get in. So it's been kind of a slow trickle.
No worries.
Are you headed somewhere fun?
I actually just got back from somewhere fun. I went for like a little hike this morning and I'm running late, so
that's
but I'm here now.
Awesome. Okay, one more. There we go. All right, guys. Well, we'll go ahead and get started. And so welcome to Dead Week. Congratulations. You made it through possibly the hardest semester of the year of ever. Although I kind of think last spring was a little bit harder. But anyway, our goal today is I'm going to go through some of the announcements and stuff just to make sure we're all on the same page as far as due dates, what's coming up, and how we're going to finish the semester. And then I've got a couple questions for us to review from chapter 12 and 13, which was new material last week. And then we'll talk about the final exam. So I do not have a climate question ready for you guys because I have a feeling that the answer would be 100% dead. I'm dead this time. So anyway, we're just gonna keep pushing through. So this is our last official Zoom Monday lecture thing. So next Monday we will not be on here. All right. So just a couple of quick announcements. I'm going to go ahead and share my screen with you so we know where everything is at. Ooh, that's not what I wanted. Cancel. I wanted to share. There we go. And get back in. All right, so we are clear down here at the bottom. So week 15. And so in this week, I went ahead and put in the submission link for the extra credit that was described in the syllabus. So that is going to be due Friday, or this Friday, the 11th, if you're interested in the extra credit. The rundown of this is basically you can do a review using the 490 Journal Club assignment, which I've attached to the submission link here. So 10 points per review, and you can do up to 2 reviews. So you can get an additional 20% of 20% or 20 points of extra credit if you so feel inclined. So there's and what you are reviewing here is actually new technology. So reproductive peer-reviewed journal articles that have been published within the last 5 years. So 2015 through 2020. And keep in mind, if you're going to do this, this is a search through Google Scholar and not just Google. So they need to be peer-reviewed journal articles. The second link in this week's folder is a video to about a 30-minute lecture on placentation and parturition. So chapter 14 of the Singer text. There is no quiz associated with this week for chapter 14 material. You will see chapter 14 questions on the final exam that's going to be due next week. So I'm not a huge fan of giving quizzes during Dead Week, so I wanted to kind of back off the pressure a little bit. And just have that information available for you to view on your own. All right, so that is what's going on in this week's Blackboard folder. So again, extra credit, due Friday by 5 p.m. And then you have access to the placentation and parturition video lecture. All right, so I do have office hours this week. If you guys are needing clarification on anything to do with the class, you can drop into my personal Zoom room on Thursday morning from 8 to 10. And that's supposed to be Friday morning from 8 to 10. Sorry, not Thursday 12/1. So Friday morning 8 to 10 as well. So Thursday is the 10th and Friday is the 11th. And my Zoom room office hour link is on the email that went out on Friday, as well as the passcodes. All right. So the last thing that I just want to kind of step through, which we'll do at the end of this lecture, is we're going to talk about the final exam. But before we get there, I want to make sure that we have a good understanding, or at least are familiar with and know where we need to spend some time studying through chapter 12 and 13 material that was new material for last week, which you guys completed a quiz on. And so we've got about 23 of you guys in the room today. And so I think we can do this together. I don't think we need to go into breakout rooms. Actually, no, you know what? I think we need to. Okay, so I'm gonna organize probably 5 breakout rooms. And so that should get 4 to 5 of you in each of that. And we're gonna start with and we'll do 5 minutes per question. And I've got 2 long questions here, and then I've got 2 short questions for the end of this review session, which we'll do which we'll do as a big group. So let me go ahead and share my screen with you, and I'm gonna pull the question up in a PowerPoint slide. So if you need to screenshot it or something, you can. And then I can also try to put it in the chat, but it might get a little bit confusing. So let me go ahead and just start with this right now. And I think I shared the right one, but now I'm not sure. Okay, can you guys see Blackboard, or can you guys see the PowerPoint?
PowerPoint.
Okay, great. All right, so here's question number 1. Okay, and so feel free to screenshot this, whatever you need to, write it down real quickly. And so what I want you guys to do in breakout rooms is kind of talk through this and get this question down, and then we'll come back and fill it in as a class. So put the following steps of fertilization in order from beginning to end. And so the steps that are just listed out here are syngamy, sperm DNA decondenses, sperm binds to the zona pellucida of the ootid, male pronucleus development, and ootid completes meiosis II, penetration to the perivatine space, cortical reaction, acrosome reaction, and the fusion protein. All right, so that is question number 1. So I'm just going to give it a couple more seconds here to either take a picture of or finish writing it down, and then I'm going to push you guys to breakout rooms. All right, it's 5 more seconds. Okay, I'm going to go ahead and stop sharing and open up breakout rooms. All right, so we're going to do just 5 minutes here, guys. So I'm hoping that this is fresh enough that it's not going to take a long time to pull all together. So I'm going to go ahead and open the rooms. If you guys will accept those invitations, I will see you back here in 5 minutes. All right, guys. Well, welcome back. I hope that gave you, well, it probably didn't give you enough time to get through the whole process, but hopefully it gave you enough time to get started on it. So, would anybody, this is the part where I need you guys to kind of help us out and speak up and not, and, or you can use the chat. What would be the first step here that would occur first? As far as the fertilization process, what is step number 1?
Is it the sperm bind to the zona pellucida?
It is. Excellent, excellent. Okay, so step number 1, sperm is going to bind to the zona pellucida. All right. And you guys can feel free to put this into the chat too. All right, what is step number 2? All right, great, Sydney. Yep, so Sydney is saying the acrosomal reaction. That is correct. Excellent. I'm just gonna put in here so we have kind of a step-by-step of what is going on in the chat. All right, what about step number 3?
Is it penetration into, I cannot say that word.
Perivitin. Yep.
Yes.
Yep. That is correct. Thank you, Marcy. All right. And then what about step 4? Did we not get this far in our discussion?
It was more like we knew the first steps and we knew like the last steps, but then the middle steps were like kind of, where does this go? Where does that go?
Got it. Got it. Okay, cool. All right.
Okay, wild guess. Syngamy? Just a wild guess.
Yeah, so you pronounced it right. So good job on that. That one's actually going to go clear to the very bottom. So that one's the last step, but great guess. So step 4, what's happening after that sperm cell has penetrated into that perivitin space?
Is that the protein fusion?
Yes, it is. Exactly. Good job. All right, cool. All right, so step 5. So once that sperm cell gets into that perivitin space and sets off, and those proteins start to fuse, what is the next step?
Would that have to do with the cortical reaction?
It is, yeah. So the cortical reaction is next. And what do you guys remember about the cortical reaction? What is its big purpose? Why does it occur?
Once the egg becomes fertilized, it doesn't allow other sperms to fertilize, which would then cause like
Yes.
mutations and stuff.
Yep, exactly. So once that those fusion proteins have been secreted, and that cortical reaction is going to take place, and that basically is going to shut down the rest of the ootid from other sperm cells penetrating the egg. Okay, so it is the zona block or the Vatine block. And so this step is really crucially important because it actually prevents polyspermy or spermatozoa multiple spermatozoa from actually entering and fertilizing the embryo. So you don't get multiple copies of male DNA. All right, cool. What about step 6? Oh, sorry. I don't think I pasted copy. Sorry, cortical reaction, control copy. I'm a little bit behind here. Oh, delete. Well, now I messed everybody up. I'm sorry. Because I'm copying and pasting. 3, 4, 5. All right, step 6. What happens after that zona block?
Is that when the sperm DNA decondenses?
It is, yep. And why is that decondensation part really important? Do you guys remember? So why was it important for DNA to condense to get into the ootid or the egg?
Does it have to do with how it can only enter the egg through those like specific cells?
Yep.
That
Yep, that's
I'm
part
trying to
of it.
remember.
Yep, so that's part of it. But also keep in mind, like we're dealing with a lot of DNA material, right? So 30 chromosomes in cattle. And so it's important for all that DNA to condense into really tightly packed chromosomes so that it all goes together into that ootid. Okay, and so we don't have long, stringy chunks of DNA getting broken off and causing a mutation, or getting broken off and not being able to fertilize the egg. So that when sperm condenses, it's really important to get that whole genetic package coming from the male that is being delivered by the sperm cell into the ootid. But once it's in the ootid, it's important for that DNA to actually relax and go back to its, I call it the long stringy form when we're in genetics. Okay. And once it's in that long string form that looks like spaghetti, that is where replication takes place. So we need it to decondense so replication can take place. And so it can go through meiosis too and get copied. All right. And so I just gave away step 7, which was kind of given away anyway. All right, so this is where step 7 is going to be where that male pronucleus is going to develop and the ootid completes meiosis 2. All right, and then Marcy already alluded to step 8, which is syngamy. All right, so that is the full process of fertilization. So I just wanted to make sure that we reviewed this because it's really important that these steps first and foremost occur, and secondly, occur in the correct order. Okay, so this is a really tightly regulated process that is one of the key drivers for reproduction even being able to take place. All right, so any questions about fertilization in our livestock species in that whole process? All righty. Okay, so I've got one more question for us to kind of work through. And it's a putting things in order question as well. And so I'm going to kick you guys back to breakout rooms. Let me show you what the question is for just a minute so we can You guys can get a screenshot if needed. Make it a little bit bigger. So the question is, put the following steps of pre-attachment development of the embryo in order from beginning to end. And so the steps here in a non-specific order are 1, hatching blastocyst, 2, early blastocyst, 3, compaction, 4, fertilization, 5, 2 to 8 cell cleavage stage within the zona pellucida, 6, syngamy, and 7, morula development. And those are just listed out. Again, that number system that I just referenced is not the correct number system. I just wanted to have it as a vocal part for the recording. All right, so I'll give you just a few more seconds to grab a screenshot of that if needed. Then we'll stop sharing and go back to breakout rooms. All right, here we go. So 5 minutes again, and we'll see you back here in just a few. And so they've all been opened back up. If you guys will just go back to your breakout room icon. Thank you. All right, guys, welcome back. Let's go ahead and kind of work through the answer to this question like we did previously. So let me make sure I don't have anybody in my waiting room. Nope. Okay. All right. So pre-attachment embryo steps. What would be step number 1 here? What has to occur before an embryo even gets produced?
Fertilization.
Yeah, fertilization. Thank you. All right. What about step 2?
Syngamy.
Yep. Syngamy. All righty. Step 3.
2 to 8 cell cleavage.
Yep. So once syngamy has happened, that kicks off the whole mitosis process. Okay, and so mitosis, if you guys recall back to genetics, or if you're in it right now with Dr. Smith, is just works kind of like a copy machine. You're just making copies of cells. So if you have 2 cells, you're going to go to 4 cells. Once you have 4 cells, you're going to go to 8 cells. And at this point in time, that zygote is just starting to get bigger and build mass. All right, so what about step 4? After that 8 cell stage, what is the next step?
Morula?
Yep, a morula.
Morula, there you go, sorry.
It's kind of tricky, super tricky. All right, so step 4 is morula development. And then step 5, keep in mind that we're continuing mitosis through this whole process. So once we get to that 8 cell stage, it's going to go to 16 cells. After 16 cells, it gets really hard to count cells. So we just call it a morula. And after morula, which is going to be several replications of cells, things inside the confines of that zygote get really, really tight. So what would be step 5?
Is it the early blastocyst?
Not quite. Not quite. Thank you for that, though. Thanks for speaking up. What if things are getting really tight within the confines of the zygote? What is going to happen?
Compaction?
Yeah, compaction. And so compaction is important here because as those cells start to compact, before it gets to the early blastocyst stage, that compaction process is really important for cell-to-cell communication. So we've got gap junctions, we've got a bunch of proteins that are on the cell surface of those newly developed mitotic cells in this new zygote that are communicating with one another. Okay, and so we need compaction to happen so that those cells are close enough that they're knocking on each other's door and telling them what they need to do. Because it's at this critical stage that these cells are starting to differentiate. So a liver cell is getting the assignment to be a liver, right? A brain cell is getting the assignment to be a brain cell, those type of things. Prior to that, in the morula stage and up to like the 16 cell stage, that those cells are considered totipotent, meaning that they don't have a specific job yet. All right, so if step 5 is compaction, then what happens? What is step 6?
Then that's the early blastocyte.
Yeah, yep, that's the early blastocyst stage. All right, and then step 7, super easy here.
That's the blastocyte hatching, right?
Yep, hatching blast. All right, so the early blastocyst stage is a really critical time frame for that embryo as well, especially if we're trying to use reproductive technology, like if we're doing embryo transfer. So we want to make sure that our embryos are in the blastocyst stage, so roughly about 7 days after fertilization has taken place. And that means that they are far enough down the uterine horn that we can actually flush them out. We can look at all that fluid underneath a microscope, and we can select an early blastocyst embryo to either put into a recipient female or to freeze. But once hatching has occurred, that freezing process or the transfer process is not going to be successful, because that whole transportation process is going to shake that zygote around pretty good. And so if there's any cells on the outside of it that have hatched out, chances are they're going to be disrupted from the bigger structure. All right, so it's really important to make sure that we have a good understanding of why an early blastocyst stage cell is important versus a hatched one. All right, cool. So that is the whole attachment process that occurs after fertilization. All right, and so keep in mind that as that process is occurring, that embryo is starting to make its way down the uterine horn or down the oviduct to the uterine horn for implantation. All right, cool. So I got a couple other questions. I want to make sure that we've got a good handle on this before we move into discussion about the final exam. And I'm not going to put you guys into breakout rooms because I think that we can just talk about these as a class. So let me share my screen again. All right, so maternal recognition, or MRP, in the sow is what hormone? And you guys can shout it out or put it into the chat.
Is it the inferion? And I forgot how to pronounce the last part.
Interferon tau?
Tau.
So thank you for participating. It's actually in the sow. It is not that substance. It is in different species though. So keep that answer on the back burner, okay? It's going to come up later. So if it's not interferon tau, what hormone could it be?
Estrogen.
F2 alpha.
Yeah, it's estrogen. It's estrogen. All right, so let me switch gears here. Okay, so this is the lecture from last week. Okay, and so when we are looking at the this figure over on the left-hand side, this is just a biopsy of the uterine tissue. Okay, and so we've got the myometrium and the endometrium here. So the endometrium is going to be this bigger kind of pink area. And so what is occurring in the endometrium is that if there is a pregnancy or an embryo that is in the hatched blastocyst stage, bouncing around here in the uterus, what ends up occurring is that the uterus is sensitive enough to pick up that extra estradiol that is being produced from that embryo. Okay, and so in the sow, the maternal recognition of pregnancy hormone is estrogen. And what ends up occurring, this is kind of a piece of nuance here, is that in response to estrogen, or the presence of estrogen, prostaglandin F2 alpha or PGF2 alpha is then rerouted into the lumen of the uterus. Okay, and so PGF2 alpha is still produced, but it's not allowed into the general blood circulation like it would be if there wasn't an embryo present or in another species. And so because because estrogen is putting up a block basically and rerouting prostaglandin F2 alpha into the uterus lumen, it's not getting into the blood supply to go to the ovary to lyse the CL and start the cycle over again. Okay, so it doesn't have the ability to cause luteolysis if there are at least 2 concepti per horn in the sow. Okay, so 2 conceptuses in the horn will maintain a pregnancy. All right, so does that make sense? Is that a little bit more clear than when we asked the question? So maternal recognition of pregnancy hormone in the sow is estrogen, and estrogen reroutes prostaglandin F2 alpha. All right, so if you guys can just give me a thumbs up in your reaction buttons or turn your cameras on or whatever, or yell at me and just tell me, yes, we got it. That's awesome. Okay, there is life, yay. All right, so let me switch gears and then ask you guys another question. All right, so maternal recognition in the cow is what hormone? And again, you can just put it into the chat or shout it out. And I can't spell, obviously. There we go.
The cow is interferon tau.
Correct. Yep. So in the cow, it is interferon tau. So let me just go back to the lecture. There we go. So in the sheep and the cow, their mechanisms, their physiology is pretty much the same. They're both ruminants. They're both going to have similar reproductive systems, etc. And so both the sheep and the cow use interferon tau as their hormone of maternal recognition of pregnancy. And so interferon tau is a really, really small protein. It's only about 20,000 Daltons. I don't expect you guys to know what that means. And it will be produced from that blastocyst somewhere around 13 to 21 days after fertilization. Okay, and so keep in mind that interferon tau, after we get that blastocyst formed and it is moving down the embryo or the oviduct into the embryo, or not the embryo, the uterine horn, it is really looking to start attaching around day 13 to day 15 in sheep and in cattle. And so that is when interferon tau will be produced from that newly formed embryo. And interferon tau will then be produced into the uterine layer and or into the uterus, and the receptors in the cells of the endometrial layer will then respond to that presence of interferon tau. And so the interferon tau will actually stimulate the uterine endometrial cells to produce a bunch of proteins that inhibit oxytocin receptors from forming. And if you recall back to chapter 9, I think I've got it here. Back in chapter 9, we talked about interferon tau, or we talked about the presence of oxytocin receptors. And if there's no interferon tau present, then oxytocin receptors are going to be produced. And once there's oxytocin receptors, there's going to be oxytocin and prostaglandin F2 alpha. Okay, but when that female is pregnant, we don't get a lot of production of oxytocin receptors. So we won't get hardly any prostaglandin F2 alpha being produced, and definitely not enough produced to get into the blood system and go to the CL and start luteolysis. All right, so take home message here is maternal recognition of pregnancy hormone in the sow is estrogen, and MRP hormone in the cow and the U is interferon tau. All right, how do we feel about those things? Those were kind of big take-home messages from chapter 12 and 13. Okay, good. I see some thumbs up. Awesome. All right. So sorry, guys. I am taking a little bit longer than I wanted to. So if you need to bounce, that's totally fine. The rest of this discussion is going to be caught on Zoom, and I'll get it uploaded as soon as I can. So what I wanted to do was just make a couple comments about the final exam. Okay. So yes, we are going to have one. Yes, it's cumulative. And it is, I've backed the intensity of it off a lot. I just cut it down from 100 questions to about 57, still worth 150 points. Okay, so the final exam will still be worth 150 points. So all your points are there. You're not going to have to worry about not having enough points. So the final exam is going to be done through Blackboard as a test on Blackboard. It will open this Wednesday, December 9th at midnight or 12:01 a.m. And it's going to close a week later. So it's going to close Wednesday, December 16th at 5 p.m. Okay, so end of business day on Wednesday, December 16th. And so Wednesday would have been the normal time that we would have taken it. Okay, I wanted you guys to have a week to be able to take this because it's an intensive class. You have lots of other classes and other stuff going on right now. So I wanted you to be able to fit that in where you could. You can expect to see quite a few multiple choice, true, false, matching, ordering, and jumbled sentence questions. Okay, there will be very few, actually, I think I pulled them all out. I think I pulled all the short answer questions out. So those will be the type of questions that you guys can expect. The good news, or maybe it's bad news, or maybe it's neutral news, I don't know, is that you're going to know your score on the final exam as soon as you submit. Okay, so it's going to be automatically graded because with the heightened timeline that the university has put on us, I don't have time to do what I would normally like to do and have you guys show me in more detail what you know and go through and hand grade it. So here's my best advice for how to get prepared for this sucker is to go back through your old quizzes, old self-studies, and study that way. Okay, so you could see similar questions pop up. You could see the same questions pop up. All right, so so just be aware of that and use those things to help you study. What else can I tell you?
Casey?
Yeah,
Is it a one and done or can you save it and go back?
it's a one and done. So you do need to take
Okay,
it in one setting. I pulled the timer off of it because I know having a set time kind of causes some anxiety. And so I think I capped the time at like 3 hours instead of 2 hours because it does make you put a time on there, but I didn't want to make it condensed to the university schedule. So 57 questions, I think, is what it ended up being. Multiple choice, true, false, matching, ordering. And yeah, you've got like 3 hours to get it done. Katie, I will try to remember to send an email. If I don't, it's because I teach all day today, and just email me and remind me. So, all right, cool. Questions? All right. I think I will also put up a link to a survey the end of this week, the first part of next week, and it'll be anonymous. You guys don't have to attach your name to anything, but I am really curious as to your feedback on the format of the class. You know, what worked, what didn't. I really do need that feedback before going into the next semester. So I hope that this has been, I've hoped you have learned something, and I'm sorry if you haven't. You have all the resources. Okay, so if you do need to look stuff up later, you can. And I'm really, really extremely sorry that this is our experience this semester. It's kind of breaking my heart as much as I'm sure it's frustrating you guys. So I am more effective when I am in front of you in person. And I know that the whole online environment thing has been really, really challenging for all of us. So, so you'll see that survey come through the kind of the end of this week, the first part of next. It'll just be a Google form. It'll be anonymous, no points associated with it. So if you guys have the time, I would appreciate the feedback there because they aren't going to get us our sets back until maybe a day before the next semester starts. So. All right, that's what I got. I'll hang out for a few more minutes. If you guys have any questions, please feel free to ask. And if not, happy Christmas, I think, is where we're at, because I won't technically see you again. So unless you get into office hours or something. So. All right.
Thank you. Merry Christmas.
Merry Christmas. Thank you. Peace out, my people.
Thanks for
Bye.
everything, Casey.
You're welcome. Thank you. Thanks for hanging in there.
Thank you, Casey. I'll see you later.
Okay, sounds good. Thanks, Zoe.
Bye.
Bye.
Thank you for